in, and it also allows things out of the cell at the proper time. The cytoplasm has all the other organelles floating around in it, as well as nutrients and proteins free floating. The nucleus, of course, the nucleus is like the boss. So the nucleus, if we were looking for an analogy, the nucleus would be like the boss of the cell. Now, let's color in some of the other ones. I left a little bit of space here. I did that on purpose because now I want to use some of the other space I left for some of the other big players. Now, connected to the nucleus, very close, is our endoplasmic reticulum. And if you draw it as a series of squiggly lines, it's a tubule. If you remember, the analogy we used was that of a conveyor belt in a factory. So a conveyor belt in a factory that transports things around the cell. Very close to headquarters, because there's a sharing of information that needs to go on. And the sharing of information between the endoplasmic reticulum and the nucleus is critical. It's vital for things to function properly in the cell. Now I'm going to put in the other players that are also involved, and that's my ribosomes. Now, ribosomes can be found in a bunch of places. See, it's smudging. Oh, I don't like that. I'm going to draw more ribosomes closer to the nucleus, and then slowly they dissipate, and that's to show the different parts of the endoplasmic reticulum. And I'm also going to show some of these ribosomes floating around. Now, I got a nice little bit of space over here that I do want to use for something else, but I need to show that these ribosomes are also free floating. I'm going to use this color to be able to symbolize those. Okay, so now I better do a little bit of labeling or I'm going to end up in trouble. Okay, so closer to the nucleus, I'm going to abbreviate this. We have the rough endoplasmic reticulum, and further away from the nucleus we have the smooth endoplasmic reticulum. Now, the reason this is called smooth is because it appears to be smooth under the microscope because it doesn't have all the ribosomes latched onto it. This part here, closer to the nucleus, is rough because it looks rough under the electron microscope. And the endoplasmic reticulum. I'm abbreviating on mine because I don't have a lot of space. For yours, I would expect you to follow proper diagramming and use the complete words. Okay? So, we got ribosomes. Those are our little blue dots. I've got a nice spot nowhere. Oh boy. Okay, over here, the little blue dots. Those are our ribosomes. Okay, so stand back, take a look at that. When you see this, now we have we've got the first 5 things that we got from the diagram just by drawing the fried egg, and we've got 3 more: the rough endoplasmic, the smooth endoplasmic reticulum, and ribosomes. So right away, if you can remember this part here, you've already passed. Okay, you've passed this part of the diagramming section. Okay, now these blue and the green structures, those are the ones that are involved in protein synthesis. So they are heavily, especially the ribosomes, are involved in building up proteins that the cell needs and that the cell needs to export to other places. What about recycling? So if you think about it, oh yeah, the name of the game is making proteins. And we need to draw our friend the Golgi apparatus in here. And the Golgi apparatus is usually seen as this weird pancake stack of pancake-like structure. It's involved in recycling, Golgi apparatus. Sometimes I like to show these little blobs. breaking off. They are also free-floating. They're associated with the Golgi apparatus because the Golgi apparatus and the lysosomes help to break apart, repackage, and export nutrients out of the cell. Those are your lysosomes. So if you've noticed, what I'm trying to do is I'm actually trying to draw the structures according to function. So I'm trying to draw the membranes, the control center, the protein building, the recycling, and if you take a look at it, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 elements. There's a few other things that were present in the reading and the diagram that we didn't have. I'm going to use red dots with little lines across it. If you don't have a marker, then you can do that. The red lines are representing kind of increase and decrease. Those are vacuoles. And remember when we were talking about the difference between plant and animal cells, if we were going to do a plant cell, this would look different. Okay, this would be a lot different in a plant cell. So everything here would be in a plant cell, except now the vacuole would have to be a little bit different. I guess the overall shape of the cell would be a little bit different too. Now we need to have the powerhouse of the cell. So what provides all the energy for maybe movement or protein synthesis, breaking down proteins? There's a structure that provides energy for that, and there's more than one in each cell. Usually we draw them like little bean-shaped, worms almost, or jelly beans. When you put these squiggly lines in it, there's a reason for that. Maybe you can learn about that if you decide to go on to study cell biology. It has to do with the membranes, and mitochondria actually have a very interesting double membrane system. So there's our mitochondria. And again, sorry for having to write sideways and stuff like that. On yours, I would expect you to do all your labeling on one side if possible, neatly on the other side. Include a figure number. I just don't have the room to be able to do that. So let's go around this circle here, kind of in the order that we did things. We have cell membrane, cytoplasm, the nucleus with its nuclear membrane and the nucleolus. There's 5 marks right there from the fried egg. And then we got the endoplasmic reticulum, rough, smooth, and the ribosomes. That's 8. We got the Golgi apparatus with lysosomes and vacuoles. Okay, that's 11. And then we have the mitochondria, which make up another 12. Okay, which make up 12 altogether. All right, so that's basically it. There's an animal cell for you. So a few things to remember. Number one, when you have to draw it, start with the fried egg. If you do the fried egg, you're going to get 5 out of those 12 possible things. And then start actually thinking about, number 2, start thinking about what are the structures? What's the purpose of a cell? Well, a cell's job is basically to build all these proteins that the body needs in order to grow and in order to conduct the life processes. So you start with protein building. Well, what does that? We got these guys in green and the ribosomes. And then we got storage, we got recycling, we got export out of the cell. What does that? Well, that gives us our ribosomes, our Golgi apparatus, our lysosomes. And then finally, we need to throw in a couple of vacuoles for some storage and the powerhouse of the cell, the mitochondria. Okay, so soak that in, get used to it. You're going to be looking at cells for a long time if you keep on studying biology. It's one of the best things, one of my favorite things that have to do with biology. Remember, cells are the building blocks of life. Every single cell can do all those 7 characteristics of living things. So I hope you enjoyed today. Today was about learning how to draw and label your own animal cell.